Okay, good morning. So if we could pull up the slides. I'm Caitilin Hamill. I'm one of the organizers of this session, and I'm also the Regulatory Affairs Director for the Cell and Gene Therapy Center at IQVIA. And our co-organizers here as well, Melanie Call. She's the Director of the Accelerating Center at Cell and Gene Therapy Center at IQVIA. And I'm very interested. I think it's going to be a great discussion about cell and gene therapy, successful strategies for your clinical development program for cell and gene therapies. Just waiting for the slides to come up. Or do I have to? Oh, okay. Thank you. All right. So this is what we're aiming to achieve. Streamlined, rigorous, well-designed clinical trials that generate real knowledge about a therapy's risk-benefit profile. So for the purposes of this workshop, that's how we define success. And this is something that I believe we really can achieve for every trial if we have intelligent, data-informed trial design, if we apply an understanding of the operations at clinical sites, if we understand the thinking of investigators, their motivations, what they do, if we understand the patient's perspective, and also, of course, the technology and the therapy being administered. But this is actually challenging to achieve for a lot of reasons. This would just be one reason highlighted here would be the increasing complexity of clinical trials overall. This is looking across all drug and biologic development. What you see is that recently, in the last about 5 years, there has been an increase in the number of procedures included in clinical protocols, increases in inclusion and exclusion criteria. We're seeing more countries, more sites. So it's increasingly complex. There's actually a decrease in the number of patients as therapies have become more targeted. And this increasing protocol complexity really comes at a cost as we have increases in protocol amendments. This is looking across all trials. This isn't specific to IQVIA trials. 60% have a protocol amendment, 48% miss their targets, 80% are delayed with respect to patient recruitment. And of course, this can derail your trial and result in missed milestones. And then cell and gene therapies are at the more complex end of the spectrum due to a lot of reasons. A couple highlighted here. Obviously, you have inherent variability of the activity of the products. The global regulatory framework is rapidly evolving, and it's less harmonized overall for cell therapies and gene therapies in particular. And it's less clear how value will be determined. And all of this has really practical implications for an individual trial. So at IQVIA, we took 2 trials, one developing a small molecule drug and the other evaluating a gene therapy, otherwise pretty closely matched protocols in terms of indication, patient population, procedures being performed. And we put them side by side and said, what's different about a gene therapy? And you can see there are more submissions to more regulatory committees. In particular, this involved Europe, and you had the GMO committees. There's more rounds of review. These protocols get a higher level of scrutiny, and that results in more protocol amendments. You have more specialty labs. You have more assays being developed. The informed consent takes longer to develop, to prepare and to approve. And the sites need more training. And also the consenting process for each individual patient takes longer as patients have to consider this really life-altering decision to commit to a gene therapy trial, and they have discussions with their family, and there's a higher rate of refusal. And all of this translates into higher costs. So for this one example, for instance, because the trial is more expensive and also involves fewer patients, you see that there was a shift from about $350,000 per patient cost to $2.1 million in this example. And so this is all a lot of challenges, and we're going to start a discussion about how to achieve streamlined, rigorous trials evaluating the cell and gene therapies. So we're going to start with a panel discussion of US-based clinical programs, and then move to the international panel, so broadening out and saying what's different when you're going into the global framework. And so I'd like to introduce Jami Norris, Vice President of Clinical Project Management for Internal Medicine and also Rare Diseases at IQVIA, who will be moderating the first panel. Whoa. Sorry.
Good morning, everyone. Really glad to be here and looking forward to the discussion. I'll just start by introducing our panelists and have them come up to the front. We'll start with Edward Worth, Chief Medical Officer at Asterias, and Peter Altman, President and CEO of BioCardia, and Dan Kaufman, who's Professor of Medicine in the Division of Regenerative Medicine and Director of Cell Therapy at UC San Diego, and Edward Worth, sorry, got confused, Felix Xu, who is Senior Vice President and Head of Advanced Therapies Unit at WuXi AppTec. So we have a great, varied panel today. We're going to cover kind of 4 broad topics here today. And we're going to start with therapy selection and trial design, and then move to patient recruitment, complex procedures and logistics, and manufacturing. So to start us off, what are some of the most important elements when designing a protocol that sponsors might not take into consideration? Dan, you want to kick it off?
Sure, thanks. So I'm coming here from, I guess the academic side, and we're really interested in getting these trial products into patients. So I guess, important design characteristics, I think, as you're, from a biotech company, designing your trials is, I think to really try and work with the clinicians, clinical team, and identify clinical teams and centers that you might want to work with, I guess, early on, and determine, what the patient recruitment, what the patient's needs are, and how you can impact that. I think when we're talking also about the manufacturing is to, consider different options that I'm sure we'll get into for cell and gene therapy manufacturing, and I think there's lots of ways to do this in the most efficient manner. But I'll say from the UC San Diego side, we often hear about this maybe a little bit late in the game, and the plan is already in place, and it may or may not fit our needs and the patient's needs, but to really try to think about those things probably early on. Other ideas?
Great. Anyone else? Yeah.
Yeah, I think one of the challenges we encountered with our program, these are allogenic cell therapies derived from human embryonic stem cells. So the cell therapies are cryopreserved in the vapor phase of liquid nitrogen. For spinal cord injury, they're administered right in the spinal cord via dedicated device and surgical procedure. The takeaway of all of this has been that site selection, qualification, and opening has been almost a continuous process that has taken far longer than I anticipated. I would say on average, probably takes us 12 to 18 months to open a clinical site. And it's a combination of all those factors, a combination of some of the steps that have to be done consecutively as opposed to concurrently in terms of budget and negotiating budgets and contracts, for example, have taken an inordinately long time at many sites. Some sites won't do that until you have IRB approval. Others require you to have the budget contract done before the IRB will even review your protocol. So they tend to force you to do those things consecutively. That often limits when and how you can, for example, train your surgeons on the injection procedure, train the BMT lab on the dose preparation of the cells for administration. And so when you just add up all of those timelines, again, it ends up being on the order of a 12 to 18 month process to get a site open for recruitment from the time you formally start working with them at the site qualification visit stage.
We've had similar experiences, but we've protocolized the whole process so that the sites know exactly where we're going and what the steps are, and then we try to bird dog it going forward. We're involved in delivering today. Our lead program is an autologous cell therapy for the treatment of ischemic etiology heart failure, and it's in a unique clinical trial setting, an IDE-PMA. I think that the thing that we and others haven't contemplated before is who controls the patients? You can have a PI for the trial that is very motivated, and they may be a great leading cardiologist, even the chief of cardiology at their centers, but the heart failure group controls those patients. And so thinking through of the whole concept of building a clinical team and making sure that you have that clinical team at a site with principal investigators, co-investigators, and that everybody who is involved in interacting with that patient is invested in the clinical trial and understands the risk-benefit equation for these patients. That's our experience.
Okay, great. What are some of your thoughts on the kind of, obviously we need complex protocols to a certain extent. What are your thoughts on the patient and site burden that these complex protocols kind of, create barriers for sites? Is there any thoughts you have there or experience with, very, very complex protocols or ways to get around that at a site and patient level?
I think part of the complex protocols are just a byproduct of the therapies that we're developing. So cell and gene therapies, it depends a lot, obviously, on how they're administered. If it's a simple and straightforward IV administration, makes it a little simpler. If it's something that requires a dedicated elective surgical procedure, for example, with a highly trained and qualified team just for the administration, that tends to make the protocol more complex. If it's an allogeneic cell therapy such as ours, you have to layer on immunosuppression and immune response monitoring. We have an oncology program, Immunotherapy program, where we're also monitoring for immune responses, but on the pharmacodynamic efficacy side, as opposed to an allogeneic cell therapy where we don't want it to be rejected. So a nice thing there is some of the assays are similar, but for different purposes, but that adds a layer of complexity. And then, of course, your outcome measures. So a lot of the indications that we're developing or targeting for cell and gene therapies are orphan indications or even very rare indications where often there's not a precedent for outcome measures that FDA has previously agreed to for either accelerated approval or full approval. So these protocols tend to be more complex because you tend to bias yourself toward collecting additional outcome measures, some of which may not help you, but to help you figure out or get a better chance of finding one that might ultimately lead to an approvable decision.
So I'll make, I think when you talk about complex protocols, and these can be complex in a number of ways. So they can be, these are obviously complex biologic agents, in I think the case that all we're talking about, these are living drugs, right? And so the manufacturing is complex. The transport to the hospital and patient is complex. But just the whole logistics, as Ed mentioned about budget and contracting and all those things are complex as well. I'll say one way that this is being improved upon, at least here in California, is through the CIRM-funded alpha stem cell clinics. So if you do have a product or trial that has some stem cell, either being stem cell being administered or targeting stem cells, there are now 5 centers: UC San Diego, UCLA Irvine is a center, San Francisco, City of Hope, and Davis. So these are now essentially additional support for clinical trials that involve stem cell therapies. So we're working with a number of groups, and the Alpha Clinic can serve as that mediator between the manufacturer and the clinical team, or between the budgeting office. If it's a cancer trial, we're also working with our Cancer Center Clinical Trials Office, and really serve to help those logistics. And I think that's something, that's also, especially at an academic center where maybe resources are limited sometime, that this has really helped us work with different groups as well. So that's, I think another resource that's out there.
So the complexity that we have is less so on the manufacturing side, because our lead program is an autologous minimally processed therapy that's processed there at point of care. But we have a couple of elements that we've woven into what we call the CardiAMP, and the AMP stands for autologous minimally processed cell therapy for ischemic heart failure, in that we have a pre-procedure selection criteria, which is a biomarker panel for the patients to assess the quality of their cells. And we may come back and touch on this more. But the issues that we have on that front is when patients have their cells assessed for an autologous therapy, there's great concern if they don't make the bar and they're excluded based on that exclusion criteria and the question of what's wrong with my cells. And so that involves some counseling on our end to the coordinators at the sites to make it clear that we're trying to focus on a therapy that has the maximum probability of success. And then for the procedural elements, our procedure involves harvest, processing, and delivery. And these are all things that an interventional cardiologist does not do today. And so we have to interact with other groups in the hospital. We have a formalized training protocol that involves bench, animal studies, as well as clinical training. And even after clinical training, we attend every single procedure that is done, just so there's technical expertise with respect to the procedure present, because oftentimes these clinical teams, between patients, they're doing 1,000 other things. And so when they come back and they have a new patient, it's helpful for them to have those touch points of someone with expertise who they can turn to quickly, and they can build a relationship of trust with regard to the trial, and they can say, this is the same as it was last time. Even something as simple as that can really help stabilize a procedure and help the team performing the procedure execute it proficiently. Our procedure, although it involves these 3 steps that are not typically done in the cardiac catheterization suite, are in fact designed to fit into the normal workflow. So there's no time down for the investigator. And actually, after the first cases in almost every site, the investigators have said, wow, that was pretty straightforward. And it's a bit of a ballet as we're moving through the room and going through the 3 steps of harvest, processing, and delivery. But because it's been designed and thought through from how this can be rolled out in the trial, and it can also have that role ultimately should the trial be successful and hit its primary endpoint.
If I could just add to your point about supporting the procedure, I mean, there's a couple of things that are come into that, because we've had this as well. So often, again, for cell therapies in particular, and even for some gene therapies, often there's a custom-made device that is used to administer the product that requires a fair amount of technical support. It's not a device that is used frequently, as you mentioned, and so for our studies, for example, in spinal cord injury, we typically had 2 to 3 technical experts from our team in the operating room for every single procedure to make sure that the injections were performed consistently, and to aid them in all the very real-time documentation that's going on around the procedure. So that's, again, part of the protocol complexity, is there's much more information that we as sponsors want to capture about that procedure that we can then post hoc go back and say, was there something different about a particular procedure that potentially led to a different outcome, better or worse? And if you're not in the room, the operating room or the interventional lab, you're not getting that information. You're not able to provide that support and ensure that consistency. And that's really important at these early to mid-stage clinical trials. Obviously, by the time you get to a pivotal trial or phase 3, you really have to have things worked out well enough to where a lot of this can be done unsupported or minimally supported. And I think as a field, we're still working toward figuring out how to do that.
Well, we're in a phase 3, and we're there for every procedure. And we do have it worked out because it's not just this program that's in phase 3. We as a organization began with a delivery technology. And the delivery technology has been used in roughly, I think, 12 clinical trials to date worldwide that have been completed. And we support those trials with our staff in every procedure because of the delivery elements in Europe and the United States and hopefully soon Asia.
All right. So what are some of your thoughts on the advantages and trade-offs between autologous and allogenic approaches to developing therapeutics?
I can give you the manufacturing perspective on that. I mean, the difference is obviously the source of the cells. So for allogenic, the cells are unrelated to the patient's receiving them, whereas for autologous, the donor basically provides their own cells and gets them back. So from a manufacturing perspective, that's a lot more complicated. Because really, you need to set up so the hospital does a draw, then the draw needs to be coordinated with the manufacturer. Then we have to have the suite ready. Then we have to have the people ready to process that. Once the process is done, we have to do the release. Depending on the release, it may be a conditional release or a full release, depending on whether it's fresh product or not and the timing on that. Then we need to work and help coordinate to get the product back to the hospital. It's got to make sure it goes back to the right hospital to the right patient. And all of that involves a chain of identity process that you have to have in place. And for us, we need to make sure everything is labeled properly, it's segregated properly, the verification is done throughout the process, and then you have your storage and transport properly handled. And I know that for a lot of hospitals, doing this type of autologous is a bit of a challenge. That's why there's a number of software companies out there right now that are trying to help manage that. So that's what you get on the autologous side. From our perspective as a manufacturer, the allogenic is, oh, by the way, the other part for autologous is that when you want to increase the amount of patients that you have, this isn't what's known as a traditional scale-up process. You can't just add more and more patients to the suites because you can only do a certain amount of patients per suite. You actually have to keep building out more suites. And so that's a very different question that's posed for our customers as to how they want to handle the build out and the increase in the patient population and how they want to look at it from a commercial perspective. Allogenic on the flip side is more your traditional batch manufacturing. It makes things a lot easier for us. If we want to grow the business, we can, if we're manufacturing the process in 200 liters and they need more, we can go to a 500 liter or we can go to 200 liters. You can scale up in the same suite. So from the perspective of how do you help the customer as they're going to late phase and commercial with the number of patients they're doing, it's easier to do allogenic. I would probably say the one other difference between them is on the release, at least for autologous, we tend to do less in the way of safety testing than we do with allogenic because allogenic is going to be used with much more different people. And so you're going to test more, especially on the viral side for that. So that's at least from a manufacturing perspective, how we look at the difference between autologous and allogenic, because we deal with both of them right now.
I'd like to dive in, Felix. So completely agree with everything you said, except there's another category, which is the autologous minimally processed. So, and I'll use by way of explanation, there's a CD34 cell therapy that's been advanced for cardiac indications. So selecting the CD34 cell after 5 days of GCSF, then the patient undergoes apheresis, then the cells get sent to a centralized facility for selection from the apheresis product, cryopreserved after lot release testing and returned to the cardiac catheterization lab. So that's selecting cells and manufacturing them in an autologous setting. Baxter Healthcare was running a pivotal trial on this not too long ago and stopped primarily because of the costs associated, we believe. What we've done is with this pre-procedure selection criteria, with a diagnostic approach, we select patients who have very high CD34 counts as one of our biomarkers. And then the therapy is a procedure kit to isolate out those cells and deliver it. And so the manufacturing for this autologous therapy becomes a device manufacturing process with remarkably low cost of goods. And it, at the same time, solves the two primary problems with respect to an autologous cell therapy. And that is patient-to-patient variation, because it acts as a, if you will, a quality control. And it also impacts the cost of goods and greatly simplifies the logistics. We also have, though, patients who don't qualify. We have an allogenic cell therapy and fits into the category that you have described. So in our hands, with the batch release, even for the autologous cell or the allogenic cell therapy, the allogenic will be far greater cost of goods than what we have in our initial autologous therapy. So there are variations on these themes. I just throw that out.
You're exception to the rule, then, is what I would say. So.
Yeah, I think the autologous minimally processed is definitely the exception to the rule for most allogeneic or autologous cell therapies. Just to give you an example, we have two versions of the same dendritic cell immunotherapy. One is autologous and one is allogeneic, derived from pluripotent stem cells. And really, our experience echoes some of the points you made, Felix, which is on the autologous side, it was this immunotherapy was tested in an AML and an open label trial. And the logistics were very cumbersome. They are getting better as some of the release testing, for example, gets better. So PCR testing for mycoplasma, for example, now has really sped up that particular release assay by a significant amount, going from weeks to days or less. On the allogeneic side, it depends again on your cell therapy. If it's a cell therapy that requires durable engraftment of the cells, such as for heart failure or for spinal cord injury, Parkinson's, what have you, immunosuppression is a huge potential issue. The jury's still out in the CNS. We use a short course, low dose of immunosuppression, which seems to be preventing rejection of our cells in the spinal cord. But again, it still is very much uncharted territory. The heart, in contrast, is completely not immunoprivileged, and it's a big problem. We've had 2 advisory panels in the past with cardiologists where we were targeting late-stage heart failure patients who, by definition, many of whom are already immunosuppressed because they have heart failure. And most of them likely could not tolerate additional immunosuppression beyond that. So if you have a heart program that's allogeneic, the big question around immunosuppression. So there's some issues both ways here. Clearly, in that situation, going with an autologous approach, as you guys are doing, makes an awful lot of sense. So there are a number of issues around both. And I think the jury is still somewhat out as to how can we efficiently bring down the manufacturing and cost of goods for autologous cell therapies and the speed of production, for example, versus the potential for really dramatic scale-up long-term for allogeneic, but still with the unknown around immunosuppression for many of these.
I can add one more comment. I think these are all really excellent points and kind of cover the spectrum that's out there. I think in one other consideration between autologous and allogeneic, especially in the cancer immunotherapy, because obviously we all know about CAR T cells has been a tremendous success, and we'll hear about that. But from the time of patient evaluation, these are autologous products. And I can say this because I'm a clinician as well, and I have patients who I want to get these therapies. It can take several weeks to months from the time that it's determined that they need, say, a CAR T cell therapy for their cancer until they actually receive that. And not every patient is going to be able to have their disease under control over that time and be fit enough over that time to get that therapy. So I know, as was said, everybody's working to improve the speed of manufacturing and such to get that time down. But we're also working on, I can say, my own research and clinical interests. We're making a allogeneic natural killer cell-based immunotherapy. We collaborate with Therapeutics on this, and this will be a quote-unquote off-the-shelf cellular immunotherapy for cancer that will be a frozen product that can be stored at sites and administered when the patient needs it. And so we think that delay of weeks will be avoided in those cases, and I think there are patients that are going to benefit by being able to get cells to them more rapidly than the autologous product. And I think I'll echo all the things that were said about the scale of manufacturing and such. For an allogeneic immunotherapy product, these are being scaled up in multi-liter bioreactors, and presumably the cost of those therapies will come down as well. There's also, with regard to the immunosuppression, the allogeneic products are certainly an issue we're now talking about comparing, say, CAR T cells, autologous, versus CAR NK cells, which are allogeneic. The allogeneic issue may be a benefit as well in that we can give repeat dosing and as a way to more precisely dose these products. As opposed to the CAR T cell, it's a one-time product. The patient has it, essentially for the rest of their life. If there's off-target effects, that's harder to control than something that might have a survival time of a few weeks in the patients and can be redosed as needed. So I think these things are all sort of moving forward as well.
Can I do a follow-up for Dan? I think you hit on a really key point there, that at the end of the day, what's most important is what minimizes the risk and maximizes the efficacy for the patient, regardless of auto or allo. In our past, we had the pleasure of co-sponsoring trials comparing autologous minimally processed to autologous culture expanded, autologous culture expanded to allogenic culture expanded, and then even dose response trials for allogenic cells, all in the same setting of ischemic heart failure. So we feel we're going forward with good information from small trials, granted, that provide us guidance on that sort of risk-benefit profile, and then enables us to consider some of the broader business issues on why we're doing things the way we are. But I think the risk-benefit is the key issue for all of these therapies.
Great, thank you. All right, if we move briefly to patient recruitment, what are some of the biggest challenges that you've encountered in recruiting subjects into a clinical trial, and how did you go about addressing these challenges?
Well, for an acute indication or subacute indication like spinal cord injury, the biggest challenge for us has been the rapidly changing standard of care in the U.S. in particular, in which 25, 30 years ago, for example, used to be a fairly small number of very recognized centers of excellence where many of these patients would be transported to and treated. And that's changed dramatically. There are now close to 500 level 1 and level 2 trauma centers just in the U.S. And many, many centers now are receiving these patients from the ambulance or the helicopter and treating them. And then they go off to wherever, home, a nursing home, rehab center, what have you. So finding these patients in a timely enough fashion to be able to get them into an acute trial that has a relatively tight recruitment window is a huge challenge, and it's necessitated really opening a disproportionately large number of sites relative to the number of patients we're trying to recruit. So for acute indications such as that, where the standard of care has changed, it's been a huge challenge. Obviously, chronic indications are much easier because you have more time to recruit these patients before you're going to administer the investigational product.
One observation of mine is that in certain areas there seem to be a lot of companies going after the same type of patients. And I do think that in talking to at least a number of our customers, they don't always consider the fact that they're all competing for the same customer and the impact that that's going to have on the recruitment of the patients. But we do see a lot of times that most companies don't hit their recruitment goals in the time they're trying to do. I think part of that's the complexity of just setting up the trial, but I think part of that is the competition for the patients themselves. So that's just a point I make because that's an observation that I've seen. If you take, I'll throw an example, CAR-T. The number of patients going after people with non-Hodgkin's or leukemia or something, there's only so many of those patients, but there's a lot of companies that are opening up in this space. So I do think the competition for the patients is also going to affect the recruitment.
Yeah, I think that's a good point. Is again in the cancer space is I can again say on the clinical side, within a center we try not to have competing protocols for the same patient population, but it's also as somebody who again does see patients, it's also knowing the field is moving so fast. There's so many other options out there. There's trials continually to open and close, and I don't think this is a problem that this group is going to solve. But it's communicating with the clinicians to what is actually open and available, even at your own center, is sometimes changing on a day-to-day basis, and just developing the database and such so that the patients and physicians know what's available is also a key issue. But that's a little bit further downstream, I think, than how the products are designed, and it was talked about before as well about inclusion-exclusion criteria and what can be done to make this more available for patients when possible. Would obviously also help recruitment.
And on our end, I think this is something we talk about daily because time is money, and a faster enrolling trial is valuable for the organization and for the clinicians involved, and even for the patients population who we're treating, because it'll provide a result sooner. So From our end, I think the key things are communication with all of the people at the centers. And some centers have it solved and work in a team-based approach far better than other centers. And then the other element is making it as easy for them as possible. And so, there's posters on the wall of the heart failure clinic, there's handouts for the patient, there's an online patient recruitment portal. We do targeted Facebook ads around the country that then feeds into that portal. And that provides, then we even have a centralized nurse coordinator who pre-screens the people who respond to those ads, and then will provide candidates to the very busy and hardworking coordinators at each of the sites, and just make the threshold a little bit easier for them, and then interacting with both the interventional cardiologist and the heart failure clinicians who are typically the lead in the programs, and making sure that they both understand every element of the trial. And even you can have that meeting once, and then 3 days later they pass a colleague in the hallway who asks them a question they don't have the answer to, that can impact enrollment. So making sure that you're always answering their questions and sharing data with them, we have found is really the best course.
Maybe if I could add one more comment to the points you guys made. For our autologous immunotherapy program, where we were looking at launching a late-stage trial in AML, high risk, over 60 patients with AML, we worked with a CRO to do a feasibility assessment, which I highly recommend. It was extremely informative. We learned, for example, that the enrollment kinetics across sites, we looked in both North America and Europe, for example, were much lower than you might have guessed. It also showed that it would be, frankly, impossible due to competition for patients, actually, to enroll an AML trial with high-risk patients solely within North America. If you wanted to enroll a trial that was going to dose, on the order of 100 to 200 patients, for example, which isn't a huge number, you absolutely would need to enroll in both North America and Europe at a minimum in order to meet the enrollment timelines that we were targeting. So another key point there, and again, it echoes the point about competition for patients.
Great. So from a patient perspective, what do you think are the biggest reasons that patients refuse to enroll in clinical trials?
In our setting, we actually have, our clinicians tell us that the patients are very excited about being in this trial. They've not had any experience like this in heart failure trials, because many of the therapies are invasive, or the drugs are, don't have some of the sexy appeal of what we're doing here in this room. But one of our principal investigators, a very distinguished gentleman, has said that one of the problems that they've seen in a center in Florida is that, there are patients who, well, why should I be involved in this trial and have a 40% chance of not receiving therapy when I can go to Mexico and get these cells infused in my vein in a day and come back? And so that's a real problem. I mean, I just received a postcard for stem cell therapy, in my knee in the Bay Area. So I think the unregulated approaches, will consume patients for some of these therapies.
I think for us, to again, to echo that point, when you're participating or contemplating participating in a trial, if it's, for example, a pluripotent stem cell-derived cell therapy, where you still have a lot of language in the consent form about the possibility of a tumor or ectopic tissue or what have you, where those points aren't made when you go to one of these, for example, for-profit stem cell clinics, all you got to do is pony up the cash, basically. And if you can afford it, you go down, and it's a pretty quick procedure. Whereas, obviously, participating in a clinical trial, you might be randomized to not getting the cell therapy in the first place. You have a long list of risks that are detailed in the consent form. Also, long-term follow-up. So our ongoing trial, for example, right now stipulates per FDA 15 years total of follow-up. A huge commitment. Granted, only the first 5 years entail in-person visits. After that is phone follow-up. But it's still just very daunting to think about a 15-year commitment to being followed in a trial.
Yeah, no, I think these are good issues. I don't have much to add. I can say from the clinical side, I mean, most patients are actually very eager to participate in clinical trials if they're available. And so it's also, like you said, it's advertising or making patients aware of what's available being referred perhaps to tertiary medical care centers because, physicians like to take care of their patients and they don't necessarily want to refer them to somebody else. So, for something like cancer, maybe heart failure, things like that, to have them seen at a specialty center where there are more options. And I can't emphasize enough about the problems of these for-profit, quote-unquote, stem cells clinics, which will literally offer therapies from A to Z, Alzheimer's to, I'm not sure what Z is, but, with the same therapy. And, I think there's a lot of confusion out there still with what these are, and these seem to be growing at almost exponential rate. And it really does create a lot of confusion for what's beneficial treatment.
All right, so we'll go ahead and spend a few minutes and move into manufacturing. With cell-based therapies, it is possible to move from a research lab into a proof of concept phase 1 study using an academic GMP manufacturing facility. What has been your experience with these types of first-in-human trials? And what are your thoughts on taking this stepwise approach to product development?
Maybe you have good experience.
I was just going to comment on the pluripotent stem cell, which is somewhat unique, so maybe there are different experiences here. But on the pluripotent stem cell side of things, back in the art days of Geron, which was, again, the first company to delve into that space, the manufacturing protocols were very rudimentary by today's standards. Materials that we use, such as the extracellular matrix, was animal-derived, number of animal-derived and undefined components in the culture media, for example. And cryopreservation entailed a thaw and preparation step prior to administration. What we're seeing now in the field, which is good and very encouraging, is on the manufacturing side, there's much better availability of animal-free components, for example, recombinant human laminin on which to grow cells. So I think we're now sort of in second generation of cellular differentiation and manufacturing protocols. We're also, I think, on the cusp now of more routinely being able to do thaw and inject. So the availability of the cryostore media, just to call them out in particular, for example, are increasingly becoming accepted by FDA for thaw and inject formulations. And I think that's really critical because for non-oncology therapeutics, asking a BMT lab to do your thaw and preparation can be quite challenging given their overall standard clinical workload and any oncology clinical trials they may be supporting. So getting to thaw and inject is an important step forward, and I think we're on the threshold of pretty good big advancements in that regard for pluripotent stem cell-derived cells.
Yeah, so I'll just add, I'll plug for the advantage of working with academic medical centers. So part of my role here is we do now have our GMP facility set up and running here at UC San Diego Advanced Cell Therapy Lab. There's information on the table for the Sanford Center outside as well. So we are really now working with academic groups as well as some small biotech startups to translate their cell products through the GMP lab and into clinical trials. And I think, we are a small center. We're really designed to do manufacturing for phase 1 and maybe phase 2 trials. We don't want to do, large-scale manufacturing for phase 3 and commercialization. But I think really, especially working with academic investigators who really, I can say, from my own experience, aren't familiar with all the intricacies of doing GMP cell production, it provides a little bit more personal service to get through the process development and to get those products made and into the clinic. And so I think we've really now begun working with several groups who have seen an advantage to working with us as opposed to, I'll say, other larger groups for the early stage trials and cell production. And, it's a little bit probably more efficient to get that done, a little bit, again, more personalized service, as well as we can connect directly to the clinical trials group, to the clinicians, whether it be cardiology, oncology, neurosurgery. We're going to start treating autoimmune disease. So, we have all those people right here, and it's essentially one-stop shopping.
So I'll make a comment from that, from our perspective, and this isn't a broad, well, this doesn't apply to everybody, but we do find at times that when we do get, they do the phase 1 at the academic center, it comes to us. Sometimes from a timing standpoint, it actually takes longer to get where they want to get. And part of that is because what we do get at times is less representative from the academic center than what's going to be in the manufacturing. An example would be if you have an autologous process, because they're doing it at academic center, they haven't really worked out the sample collection procedure that you want standardized across all the sites, as an example. The prepping, the shipping procedure might not be worked out. The QC may be different done at the academic center than what you want to do on the commercial when you're looking that way. I think there's considerations on final container that aren't necessarily worked through. So I think there's pros and cons to doing both of them that way. I will say our experience has been on a number of occasions when we have gotten them that we ended up having to do a lot of these things. And we think it might have actually taken longer than if they just started with us on phase 1. We're willing to work either way. I think it works because when you look at the centers are going to decide what they're going to do based on a couple of factors like what's my budget, how much money have I raised. Part of it will be how many people do they have on staff, who do they have that can do the work versus what they have to outsource. So I think there's a lot of different things that happen, but I thought I would at least throw the other viewpoint on. I think there are people who work with academic centers that's fine. I think people can also start with a manufacturer or CMO. You can do it either way. I just threw up some reasons on what's happened for both sides of us.
And on our end, we have manufacturing issues and logistics that impact the diagnostic element, the devices involved in the autologous therapy, as well as the delivery that's used in every single therapy we're helping with. And then we also have our allogenic therapy. And so on the allogenic, we've decided to bring it in-house, actually, and do the pilot manufacturing ourselves because we have core competency there, but also because the contracting and the economics and being close to the cells is valuable for dialing things in and getting it done on our timeline. And the cost to actually put in a facility is actually quite modest if you have the experience in-house for a single, early therapy development effort.
Great. Any other considerations?
Just one, you think, for a lot of cell and gene therapies, what we're hearing from our regulatory consultant now, who's was in CBER and now is consulting full-time, many programs, the CMC is lagging behind the clinical development. The trials are racing ahead of the CMC development, and more products now, unfortunately, are getting to phase 2, even phase 3, or even pre-BLA, for example, and they're not CMC ready. She just mentioned it in a meeting that I was at NIH last week. There are 5 cell and gene therapy products right now that are at the pre-BLA stage. FDA has said on the clinical side, the data are compelling enough, they would absolutely approve these. Unfortunately, all of them are nowhere near CMC readiness for approval, and this is becoming an increasing issue. So FDA is getting more aggressive about holding up early stage programs, even at the IND stage, if they feel that the CMC side of things isn't as ready as it should be relative to where the clinical program is. So just a word of caution there looking ahead.
All right, great. Thanks, everyone, very much. I think we're out of our time. It's been a very engaging discussion. I want to thank the panel very much for participating.